Welcome everyone to PDHC's Peptide Synthesis Innovation webinar. My name is Hanson, and along with a few others in the PDHC, we're helping to organize activities around the peptide synthesis innovations, which is one of the three core initiatives within the PDHC. This initiative is focused on pushing forward the topics and advancement of enabling complex peptide synthesis, address cost and scalability from R&D to commercial scale manufacturing of peptides, and process efficiency and green chemistry. So in that vein, we have this webinar series where we want to highlight key advancements in the peptide synthesis innovation arena. We have three amazing speakers here that you can see with very interesting talks. Each presentation will be about 20 minutes. Followed by a 5-minute Q&A. So while the speakers are presenting, if you have questions, please go ahead and type those into the Q&A section of the webinar. So you'll see a Q&A button at the bottom of the Zoom, and you can type in your question. So when you type in your question, please include your name, company, and your question in detail. Fernando will be moderating the Q&A section and going through those questions as the speakers are presenting. And at the end of their presentation, Fernando will be asking those questions live to the speakers. We do want to stay on time. So for any questions that we can't get to live, the speakers, they might be able to type in the answers into the Q&A chat after their presentation and Q&A section. Okay, so without further ado, first talk is from Don Wellings. Don is the founder and managing director of Spheritech and the lead author in a recent Nature publication that was just published in February on water-based coupling of amino acids for SPPS. I've read the publication, super interested in this topic, and looking forward to his presentation. So Don, if you want to take it away.
Okay. I shall share. Hopefully everyone can see this now.
Yeah, we can see it.
Okay, so I first have to thank you for allowing me to speak at this webinar. First one I've ever done, so hope everybody can bear with me. I'm talking on April Fool's Day, and it isn't a joke, it is real. And these are my three, well, the three principal leaders of the groups that have got involved in this project. So we've been working on making peptides in water for almost 3 years, and I've been working closely with John Wade, who's a long-term collaborator over several decades, and more recently with Morten, who joined the team. Morten and John call us the Dream Team. I'm not quite sure that's right, but I'll move on. As Hanson has already mentioned, the whole of this is based on a paper that was published back in February. It was accepted last year for publication, which involves a whole group of people, which I'll summarise towards the end of the meeting, towards the end of this presentation. But we're talking about solid phase peptide synthesis in water. So the process, as everybody knows, is simple, it's repetitive. There's nothing complicated about it. It's established methodology going right back for 60 years. Well-established chemistry, whether it's coupling with carbodiamides, uranium complexes, etc. And it's a reliable system, hence it's automated. But it does have its problems. The main thing that we're focusing on here is its sustainability. The process is never 100% efficient. It's not sustainable. Now, measuring sustainability is also not easy either. We're making peptides in water, so it's still a solvent. If you look at the way people tend to measure sustainability, they use the process mass intensity, which is basically how much reagents you use to make a quantity of a product, whether it's a peptide, whether it's a small molecule, whatever. When it comes to making peptides by solid phase chemistry, the bulk of the PMI comes from the solvents. Okay, so we're still using water, it's a solvent, but it's a little unfair to treat water as a solvent. We can take that water and we have purified it and reused it. So it's very simple to reuse and reapply for washing cycles further down the line. This just summarises what would be typical in a normal peptide synthesis on, say, polystyrene. People do have a thing about oxymer pure these days, but obviously we're moving into water. The whole thing around here actually comes from a strange observation that I made a few years ago. We were actually looking at green chemistry, not necessarily using water. And I discovered by accident, purely by accident, that if you take one equivalent of N-methylmorpholine, a water-soluble base, to one equivalent of FMOC amino acid, you get a clear solution in water. So this first picture here is actually FMOC phenylalanine at 0.8 molar concentration, which is actually, yes, by weight. It's 31% weight volume. So quite an interesting observation. If you dilute it, 0.4, still nice and clear. You dilute it to 0.2, you can still, it's starting to get hazy. If you go beyond 0.2 molar, you form micelles. So it gets quite cloudy, and the lower you go, the cloudier it becomes. So this isn't a micelle. So at the higher concentration, it is a true solution. So a little odd, and try and explain it as we go through. But we've taken all of the FMOC amino acids, and if you use water-soluble bases, the N-methylmorpholine is our favoured. Triethanolamine is another good one, and hydroxyethylmorpholine is also a good one. And like I say, I'll explain why we chose these shortly. But with N-methylmorpholine, one equivalent of N-methylmorpholine to one equivalent of FMOC amino acid, and you will get a clear solution at concentrations of almost up to 1 molar concentration. Right the way down to 0.4 molar concentration. The ones that stand out as not being soluble, not surprisingly, are amino acids with tritol groups on. So cis-tritol, for instance, is insoluble in all of these bases. However, you don't need cis-tritol. We can use cis-ACM, and there are other protecting groups as well. So you can see that even, FMOC glue tertiary butyl is totally soluble at these concentrations. We like to work with FMOC threonine, FMOC serine unprotected. So we're looking at minimal side chain protection. If you're coupling in water, you don't need to protect the hydroxyls. There is no purpose to it. So what's going on here? It's a hypothesis, but maybe we have a very strong ion pair, and we have a hydrogen bonding between the oxygen in the N-methylmorpholine in this case, and the carbonyl of the urethane protecting group. It is interesting in that all urethane protected amino acids appear to be solubilized in this way. So I'm not just talking about FMOC amino acids, BOC amino acids are also solubilized, Z amino acids are also solubilized. So it's a hypothesis, difficult to prove or treat, but whatever is happening, we have a water-soluble complex. Okay, so how do we do things? Coupling in water has also thrown us a little bit because, okay, first we discover we can dissolve amino acids in water. Then we discover that we can't actually couple them efficiently for two reasons. One is the existing procedures, so things like DMTMM, EDCI by itself, the water-soluble carbonamide, do not couple in water efficiently. So what we had to do was develop another coupling agent. So what I did, I had the idea of using EDCI, HOBT and forming an adduct between them. And this is the adduct that we formed between them. Interestingly enough, this adduct forms quite rapidly. It's certainly formed within 10 minutes or so. And it's stable in water. It's stable in water for very long periods. And you can see from the NMR data there that 10 minutes it's formed, it's stable. We get some hydrolysis to the urea, of course, as you might expect, but 30 minutes it's stable, 60 minutes it's stable, and even overnight at minus 20 degrees centigrade, it's stable and it works. So effectively what we're doing is we're forming HOBT esters in situ, and we've shown this both by HPLC and by NMR. So it forms HOBT esters, which are great coupling agents. And actually, under these conditions, at pHs between 6 and 7, they are remarkably soluble in water. Okay, we then hit another problem. Some people are aware of our peptide synthesis support Spheritech, which is based on cross-linked polyepsilon lysine. Now, a normal version of this is cross-linked with sebacic acid, which is a C10 biscarboxy fatty acid. And we discovered that this actually wasn't hydrophilic enough to do peptide synthesis in water. As soon as you couple an amino acid, it collapses. So a simple change of cross-linking from sebacic acid to 369 trioxoundecanoic acid, a biscarboxy PEG, solubilizes and solvates the product in water. The pictures on the right-hand side there, you see are a macroporous hydrogel. We use a porogen to create macropores in the structure, and this allows even better mass transport throughout the particle. The materials are very mechanically robust because they're very highly cross-linked, but fully solvated in water. This gives you some idea of the solvation. So we've taken 0.5 gram of material in each of these cases, and you can see you can still use this material in DMF. It'll swell to 6 mils per gram in DMF, swells more in water, swells a little bit more in methanol. But one of the big advantages of doing peptide synthesis in water is the ability to use chaotropes to reduce potential aggregation. So we're also assembling peptides using aqueous urea. So 50% weight aqueous urea swells the resin even more. So this is a polyepsilon lysine cross-linked resin. So we then have another issue. We want to get away from piperidine. We need to get sustainable. Lots of people have reported on the various bases that are used, and we focused on one base, which I'll come to shortly. But what we need to do is make sure that we remove the dibenzofulvene. Dibenzofulvene can cap the amine of the growing peptide chain on the solid support, and we have seen this in the past. So sequestering the dibenzofulvene is very important in a batch-wise reactor. Under continuous flow, it's less of an issue. So what we did is we went back to what other people have done. We've looked at morpholine. Morpholine we chose for a good reason. It is sustainable. It's made on a very, very large scale, multi-ton scale. It is also biodegradable in sewage sludge. So it has some advantages. It's a very green base in that respect. The problem with these things is we have to sequester the dibenzofulvene, the DBF here. And like piperidine, it is very slow to sequester the dibenzofulvene. As you can see here, we have a 5-minute, 30 minutes, and 60-minute reaction with just morpholine on the left-hand side. And on this side, we have a reaction adding ferric chloride. Now, ferric chloride is interesting in that it catalyzes the addition across the double bond in styrenic polymers. So it's quite an interesting one. So I decided to add ferric chloride to my morpholine. This isn't novel. It has been reported many, many years ago, but seems to have slipped under the wire somewhere. And what we see here is a very rapid sequestering of the dibenzofulvene. So within 30 minutes, it's virtually undetectable. And similarly, after 60 minutes, it's gone. So we're less worried about dibenzofulvene when we use 1% ferric chloride in morpholine. So not necessarily solved. We would love water, but you can't do this in water because it precipitates. So to some real peptide synthesis and some further work, so you can see what we're doing. We wanted to look at using the morpholine with ferric chloride and aspartamide formation. So we chose this peptide. It's a standard test peptide for aspartamide formation. It's from a scorpion bone, if I remember correctly. We assembled the peptide in DMF using 20% piperidine and have a baseline hexapeptide concentration of 87% and aspartamide of 2.04%. So we then stress tested that peptide for 4 hours in 20% piperidine. And after 4 hours, we've reduced the peptide down to low levels, and we've increased the aspartamide dramatically. If you do the same with morpholine, so this is just neat morpholine, you're nowhere near as bad. You increase the aspartamide up to 4%, you decrease the hexapeptide down, but if you do it with 1% ferric chloride, then we're at a similar level to the starting point. So adding the ferric chloride also has an advantage in aspartamide formation. So this is where we do. So we now have a fairly standard water-based solid phase process where we use neat morpholine with ferric chloride. In between going from water to FMOD protection, we have to do a wash with another solvent. Now, we used acetonitrile. You could use isopropanol. The reason for doing this is if you have water present, you will precipitate the dibenzofulvene, which obviously causes mechanical issues and engineering issues.
Dad, I think we lost your screen or your slides.
Oh, where did you lose me?
I can still see it.
Oh, you can?
Yeah.
Okay. Hopefully everybody else can. So we're using this process to go and make peptides. So obviously we choose a simple peptide to start with, which is leuenkephalin. And you can see it assembles nicely, a reasonable purity, and the mass spec is spot on. Same again. We looked at the more difficult sequence that again is a fairly standard chosen sequence, which is ACP 65 to 74. We deliberately, because we expected this to collapse, assembled this in 50% aqueous urea. And as you can see, we still have a reasonable purity. You can see from the blown up section, you can see reasonable purity too. And the mass spec, again, is spot on as we expect. So 2 peptides in water. Moving on to something a little bit longer, a little bit more complex, we chose bivalirudin. A lot of people use this as an example. It's 20 amino acids, and we took samples out at position 11. So we have a decapeptide in HPLC trace A. Very nice clean decapeptide, crude peptide. We took a sample out at 12 amino acids, which is sample B. Again, relatively clean for a crude peptide. Taking a sample out again at the 16th amino acid coupling, which again produces a relatively clean material for 16 amino acids, and ultimately at the final coupling of D Phi, we take the 20 amino acids. By this time, we've picked up a little impurity. We haven't investigated what this is. We're demonstrating that a 20mer made entirely in water using this strategy comes out at a reasonable purity at 83%. So no development of this, just a straight through assembly. I'm going to diverge slightly, still working in water, just to remind people that way back, these 2 gentlemen, Bob Shepherd and Eric Atherton, were instrumental in moving forward with FMOC solid phase chemistry, and ultimately with column-based chemistry using FMOC amino acids. Sadly, Bob has now parted with us a couple of years ago. Eric is still very much here, and I see him on a regular basis. But if we go back in time, back in the 1980s, Eric and Bob worked very hard on continuous flow, which is where I want to move to talk about. Now, obviously, with continuous flow, you can have inline column monitoring, various techniques, but originally by UV absorption. A major issue with continuous flow was the swelling and contraction of resin during assembly. There are ways around this by having expanding columns, but that's overcomplicated. So what Eric and Bob decided to do was to use an exoskeleton to support the peptide resin. So they had a polydimethyl acrylamide resin, which was supported inside a Kieselguhr silicaceous shells of unicellular aquatic plants. So a rigid material. It was never quite perfect because that material is quite fragile in its own right, and maybe the techniques to actually encapsulate that particular polymer at the time weren't optimized fully. But we did use those, and we did sell those for many, many years, and even use them up to 15 centimeter diameter columns. but never quite perfect. If you want to look at the history, we also have a recent review which was published back in December last year, a tribute to David Craig on his 70th birthday, which summarises continuous flow-based peptide synthesis. So what have we done? We decided to move away into a totally different concept. So a new concept in solid support. People are now aware that we use polyepsilon lysine cross-linked called Spheritech. Now, we want to get more sustainable, we want to get less expensive. So we've taken materials that are readily available on a very large scale. So example, what we use as our backbone is polyallylamine. Polyallylamine is made on a very large scale for the detergents industry and for the medical device industry. Formic acid is obviously inexpensive. We use this to control the capping. We can cap with other acids. And we do actually cap with other acids for many applications. The capping controls the properties of the polymer. And we still cross-link with this bis-carboxy PEG, which is also made on a large scale and is used in the cosmetics industry. So it's in lots of shampoos, etc., and creams and the likes. Okay, so we now have a cross-linked polyallylamine that is capped to control the loading. The polyallylamine starts off at around about 18,000 molecular weight. By the time it's capped, we're up to a much higher levels, and it's cross-linked, and it behaves identically in the same way as cross-linked polyepsilon lysine, but it's inexpensive. So now, imagine taking this new polymer for peptide synthesis and encapsulating that inside a macropore exoskeleton, a little like the work that Bob Shepherd and Eric Atherton did in the 1980s with Kieselguhr, but something a little bit more sophisticated, a little bit more robust. And we've done that. So we have sourced macroporous beads. On the top picture here, you can see these are typically around about 500 micron up to about 1 millimeter in diameter. And if you look at the pore structure inside these beads, it's very open. So we have interconnecting pores of between about 1 and 5 micron throughout the entirety of all these beads, which is actually much akin to the Kieselguhr Eric and Bob used back in the 1980s, but we have a nice, rigid, mechanically robust spherical particle. Okay, so why do we go there? This particle, this exoskeleton, is manufactured on a multi-ton scale. Polyallylamine is also manufactured on a multi-ton scale. The cross-linker is manufactured on a multi-ton scale. And all of the capping agents that we use are also manufactured on a multi-ton scale. The actual capping agent we use in our commercially available material it actually costs £300 a ton. So you can see we are looking for sustainability in terms of cost and, of course, their use in water. So we have now this material. It is commercially available. I'll thank the team that I've had behind me and with me for the last few years. John Wade and Feng in particular, I've worked with for, I don't know, maybe 20 years plus. This is my team here, Josh Greenwood, Ian Thomas and Colin. Ian Thomas has been working with me in peptide synthesis since the 1980s. Over at the University of Copenhagen, we have Morten Meldal and Ariana, his colleague, who did some X-ray work for us as well. Over at La Trobe, so you can see it's quite a big team. We have Wenyi Lee. And of course, everybody needs financial support. Obviously, we financed our own work on this side. There's the Australian Research Council, Novo Nordisk Foundation to the University of Copenhagen as well, and the NHMRC that's funding the Wenyi and some of the work at John Wade. So that is the end of my talk. I'd like to thank you and open for questions. I think I'm spot on time.
Yes, thank you very much, John. So far, we have 2 questions.
Yeah.
Okay, the first one is Julio Cassi from Sen Chemicals. And Julio says, wonderful work. have you tried to test treatment of the water which comes out of the process after it has been recycled for washings? You are using again the water. You are recycling the water?
Yeah, so if you take the water that comes out and you pass it through an off-the-shelf water softener, which is a mixed bed anion and cation exchange resin, then you can remove those impurities. That work is actually shown in the publication we did in February, but you can then use that water. I wouldn't use it necessarily for coupling, but we use it for washing towards the end. So you can reuse that water for some of the final washing stages, which again reduces the amount of water that you need. But like I say, I wouldn't necessarily use that directly for coupling the next amino acid, but certainly for wash cycles.
Okay, thank you. We have another question from Priyanka from KwaZulu-Natal, and she says, I am curious about the function of the ferric chloride here. And she has two questions. Do assist morpholine in catalytic DBF also to contribute the faster FMOC deprotonation?
It's not to catalyze the faster FMOC deprotection. The ferric chloride in there catalyzes the addition of the base across the double bond in dibenzofulvene. But it does appear to have a mechanism for reducing the spartamide formation that we don't quite understand yet. Does that answer both questions?
What wonderful! Can you explain what is 50% urea in the what is the role of the 50% urea in the swelling of the resin?
It's as you know, ureas are chaotropes, so they're very good at breaking down hydrogen bonding. So I think just the swelling of the resin is to break down the hydrogen bonding between the backbone and the actual polymer. But when you're assembling a peptide, which is what we used it for the ACP sequence, is to reduce aggregation during peptide assembly. We need to go further, so we need to choose some longer peptides that aggregate badly, such as the beta amyloids, for instance. But we're fairly comfortable that we can make, and we have actually made many peptides using 50% aqueous urea.
Okay.
Okay.
Thank you very much. I think we don't have any further questions, and we can move to the second speaker, Hanson.
Yeah. Maybe Don, I'll sneak in one more question. It sounds like you guys are starting to make a few longer peptides as well. From what I understand in the paper, you were making up to 20 mers. Can you speak to the ability of this water-based method for making longer peptides, maybe in the 40 mer range? That might be also more aggregation prone and might have some hydrophobic content like some of the GLP-1s.
We will ultimately investigate that. We have actually made glucagon. It wasn't as clean as we would have hoped, but that was in the early days where the technology wasn't so well developed and we hadn't changed our solid support. But the major peak in that glucagon was glucagon. And that was confirmed by John Wade's group. So we have made, typically peptides of that length. I would like to investigate longer peptides. The solid support is capable of doing it. People have made up to 84 amino acids on our solid supports. But I think in the longer term, people are going to be doing fragment assemblies. So we have made fragments of tirzepatide a few times now. Breaking it down, and the longest fragment is 15 amino acids. Yeah, we would like to look at longer, but I think in the long term, it'll be fragment assembly, maybe enzymatic couplings as well.
Okay, yeah, very good. Pascal's going to be talking a little bit about some fragment conjugation in his talk.
Yeah.
Okay, all right. Well, thank you, Don. And moving on to our second talk here. So Tina's going to be giving her second talk, or the second talk. She's the co-founder and CEO of Aralez Bio, where they design and manufacture non-canonical amino acids. These provide a number of benefits from improved stability to permeability in peptides. And she's going to share how they're utilizing an enzyme platform to enable scalable production of a diverse range of non-canonical amino acids while simultaneously significantly reducing waste. So Tina, if you'd like to take it away.
Perfect, and thank you everybody for getting up to attend this webinar. It's been really fun to hear Don's talk so far and to participate in this great program by the PDHC. So I'm the co-founder and CEO of Aralez Bio, and we are interested in non-canonical amino acids, by which I mean anything that is not one of the standard 20 amino acids that you would find in proteins. And we've developed an enzyme platform that allows us to really explore these in new ways. So we've worked on the breadth of amino acid diversity, so we have the largest collection of these non-canonical amino acids in the world. We also have been working on using these same enzymes to scale up everything from drug discovery to commercial scale and make a process that makes really high purity amino acids, that's green, that's affordable. And we're also beginning to work on really understanding these amino acids in different ways and screening them doing these different screens, doing these computational methods, and we're finding a lot of cool stuff. Obviously, amino acids that are good for making new peptides, but there's also some that are 1,000 times sweeter than sugar and some that are blue. So this is really a totally new area of chemical space that we're just beginning to explore. And within peptides, these are really important because they're in over 60% of approved peptides. So if you look at this chart, the orange line is the number of approved peptides and peptidome mimetics over time, and then the teal line is the number of non-canonical amino acids within said peptides. And as you can see, within the last 5 or so years, that slope is getting a whole lot steeper on that teal line. And that's because while historically there was about 2 non-canonical amino acids per your typical peptide, now there's a lot in the clinic or recently approved that are 50, 80, 100% made of these non-canonical amino acids on a per residue basis. So these are becoming a lot more frequent as we're really getting good at tuning the peptides tuning the properties of these peptides. And obviously, as peptides has been undergoing a renaissance, the quantities have been getting just so much larger with things like GLP-1s, which then makes the ability to manufacture these a lot more essential. And these are going to be used at all different stages of drug development. Things like Peptidream, other screening technologies are deploying these in drug discovery. They're also used to really fine-tune atom by atom in SAR for lead optimization, and then of course for manufacturing the final API as well. So in order to make that happen, we ideally want to have a process, much like Don was alluding to, that's efficient in a lot of these different ways. We want to be able to make a variety of these different non-canonical amino acids. We want to be able to make them at scale, and we want to be able to do that in a way that's green. But I would argue that as a field, we are not really great at most of these, and about 5% of alpha amino acid space is currently commercially available. So we're really just scratching the surface. And a lot of these amino acids have only been made at milligram to gram scale, and really trying to scale them up, even at that stage, can take months and is often pretty expensive. Many of them have had a lot of trouble scaling up to larger scales as well. And on the sustainability side, most of our methods generate 100 times more waste than product. So this is really a nascent field, and there's a lot of great work that's being done in a variety of different areas to improve this so that we can continue to make great new peptides. So a lot of the common methods are chemical synthesis, which is amazing at making a really broad scope of these different amino acids. There's methods like palladium catalyst aziridine and serine lactone. Some downsides are that these require a lot of protection and deprotection steps. They're often not stereo or regio-selective, and you often have to synthesize these chiral auxiliaries that are basically as complicated as the amino acid itself. And this is really the most ubiquitous methods today. And I'm a biocatalysis believer, but a lot of the methods that exist here aren't really addressing the problem either. These are great because they're very selective on the stereo and regiochemistry. The enzymes can be really easily produced by standard industrial fermentation, but a lot of these reactions are reversible, so that's going to be hard to make a scalable approach. And some of them, such as the aminotransferases, only really set the stereo center. So similarly, you had to synthesize most of that amino acid scaffold before you even got to the point of using the biocatalyst, which isn't really saving us that much in terms of steps. So our work, first at Caltech and now at Aralez Bio, has really been around how can we make enzymes that sort of address all of those strengths and as much as possible mitigate those weaknesses. So this enzyme that I'm showing is tryptophan synthase, which natively synthesizes tryptophan. And it takes serine and indole and does a PLP-mediated beta substitution reaction in order to make tryptophan with the release of water. So this has some good process chemistry aspects to it because this is a one-step reaction, it's irreversible, it's from these commercially available starting materials, and this reaction can be performed fully in water, so we're really reducing the amount of solvent. A lot of times people will ask questions about, water of course is going to be inherently greener than a solvent, but if you needed to use a huge volume of water because you can't run these at high concentration, then that isn't necessarily going to be any greener. But we found that a lot of these enzymes can tolerate molar concentration of starting materials such that we can reduce the volume quite a bit as well. And this is doing a bond-forming reaction, not just setting a stereocenter, and the products it makes are totally antioPure. And if you look at the catalytic cycle of this enzyme, down here, this is the key step that we're talking about. This is the PLP cofactor. It binds this serine substrate, which forms the amino acrylate that serves as the electrophilic species. That's then attacked by this indole substrate in order to form that carbon-carbon bond, which then creates tryptophan, which will be released. And when we were starting to work on this, we thought that maybe this reaction could be pushed a whole lot further than what nature originally intended. And so we thought that probably this substrate 1 could be any amine that can interact with this cofactor and that can form this amino acrylate or similar electrophilic species. So for example, threonine we thought would work in order to make a beta branched amino acid. And we thought, okay, this indole doesn't have to be indole, it should be able to be any nucleophile that's able to attack that amino acrylate and is stable in water, since that's where we're performing our reactions. So we thought, okay, maybe that should work with phenols in order to make tyrosine analogs. And what we did is we used directed evolution in order to engineer enzymes that could accept broader and broader substrate scopes. So just very briefly, the idea with directed evolution is that you start with a parent enzyme, which is any enzyme that does the thing you want at least a little bit. So in our case, that's an enzyme that can make some type of new non-canonical amino acid, even if it's just one turnover, that's okay, we can start from there. Then we create a library of different enzyme variants by incorporating different mutations with a mutagenesis technique, and then we screen for its ability to make that new non-canonical amino acid or to make that amino acid more efficiently. And then we just iterate through that process until we're satisfied. So this is one of those slides that's like, long story short, a lot of pain and suffering and work happens behind the scenes here. But we really did push this so that with this enzyme, tryptophan synthase, and with others, we have been able to make a really wide range of these different non-canonical amino acids on a variety of different scaffolds. So, this side chain can be any aryl alkyl, thioalkyl, and these R groups can be most things that you would want to apply in medicinal chemistry. And that's just by virtue of really exploiting that catalytic cycle and the inherent promiscuity of the enzymes. And then we've also been doing some work on that sort of substrate 1 piece in order to change the side or the backbone as well. So obviously we can make L-enantiomer amino acids, but we also can make D-enantiomer, alpha-methylated, or beta-branched, or homologated. And so with a lot of protein engineering, we've been able to access a lot of new chemical space. And about 70% of these weren't reported before. Obviously, it's, I think, hubris to say that chemistry can't make something, but these are new amino acids that they're being reported for the first time. And this isn't just an intellectual exercise, right? This is really important because now we can begin to tune atom by atom the different functions of this peptide just by modifying its structure. So, for example, there's this cyclobutadienylalanine that's really, really great for these pi-star interactions in your peptide. There's this dihydrotryptophan for enhanced bioconjugation. And then we also have this methylene phenylalanine, which is great. It's like almost completely insoluble in water. So if you were looking at these and making a face about these chemical structures, you are correct. None of these can chemically exist. We like weird amino acids, but that's a little bit too far. So that's my April Fool's prank on you. Hopefully wake you up in the morning. But this idea is still true even if those structures can't exist. So we really can change these atom by atom and say, okay, if we incorporate a endocyclic nitrogen, then we can increase the solubility of the amino acid and therefore of the peptide. if we modify some of these side chains, you can really fine tune that interaction of your peptide with the binding interface in order to get something like a picomolar binder. We have D-amino acids, alpha-methylated amino acids that can improve protease resistance and contribute to the longer half-life of the peptide. And there's some things like this rigidification that can contribute to the membrane permeability of the amino acid and the peptide as well. So we're really excited to see how this expansion of chemical space really leads to being able to make these awesome new peptides. So we talked about diversity, but now let's talk a little bit about scale up. So with our biocatalysis, we can scale this up in a pretty straightforward manner. We try to use tarp and shovel chemistry that's pretty easy to deploy. So what we do is we take that enzyme and those two starting materials, dump it into a normal jacketed reactor. And a lot of the enzymes that we work with are from hyperthermophilic organisms that live in hydrothermal vents under the sea, which then means that they're very heat stable. So we can heat up this reactant to 70 degrees and get it to proceed pretty quickly. That way we can get to essentially quantitative yields of the amino acid production. And we've worked out conditions so that we can really get that amino acid to just precipitate out from that reaction mixture. And then you can just filter the reaction and you end up with this big old amino acid cake. You can rinse away some of the residual starting materials or any other organic solvents. And at that point, the amino acid's typically about 97% pure. That's good for a lot of applications, but obviously since we're talking about peptides, we're also usually going to do then a protection step, and this is just a standard FMOC or Bach protection or whatever your favorite protecting group is, and then we'll just recrystallize from there in order to get to a really pure amino acid. And when we started this company, we could make 1 gram of about 30 different amino acids, and I thought it was so cool being able to do that on the Biotage. But now we've scaled this up from milligrams to kilograms, and now we're working on tons of amino acid. And so far the biocatalysis has been really scale friendly, and that you can do this exact same order of operations at in a round bottom flask as in a really big 1,000-liter reactor, and seems to be working pretty straightforward. And tying this into the sustainability, as we have a process that's more efficient, then you can see here that this purple line is us, and this other bar is the chemical methods that are commonly deployed. This process just ends up being a whole lot greener, and that's just by virtue of having this more efficient process. So if you look at these various chemical steps, the chemical method is typically 5 to 10 steps, whereas to make the amino acid is 1 step with biocatalysis, and the time is a lot less, but also so is the yield, right? Even if you have a 95% yield on every single one of your steps, we all know that the more steps you do, the harder it is to get a really high overall yield on your process. And as I mentioned, being able to run these with the enzymes at molar concentration really reduces the reaction volume, which cuts down on a lot of that waste as well. And we don't use any hazardous materials in the biocatalysis, so as Don was alluding to, sort of what that waste is, we think matters as well. And so that really then ties into having this improved mass index, improved life cycle energy, and this reduction of CO2 overall. A lot of the contributions for the energy and CO2 do come from that removal of solvents, which is a really major contributor. So to tie this back in to peptides, there's this really great paper by the GCIPR where they took a lot of different peptides that have PMIs reported and looked at the different variability of how those perform and how those process conditions can change it. But they didn't include any of the PMI from these non-canonical amino acids. So that's data we have. This is obviously not published, but if you look at that paper, the average reported PMI was 874 for a typical commercial peptide. What we show here is the PMI for the amino acid synthesis. So all of this is kg per kg, so kg of peptide or kg of amino acid. And with us, we get, depending on the product we're making, 10 to 40 PMI. I get asked a lot about what is the contribution of the enzyme production to the PMI, and that typically is +2, so it's not really a major contributor. And then what I'm showing here is 3 chemical methods that are commonly used to make these non-canonical amino acids, and for those we're assuming 100% yield at each of these steps. And the PMI also varies pretty substantially, but anywhere from 220 to 2200 PMI. And so the contributions of these non-canonical amino acids on the overall sustainability of the peptide really cannot be overlooked. And especially as we're using more and more complicated non-canonical amino acids and peptides, then it becomes a lot more substantial as we want to see this reduction in our overall sustainability aspects. And so with that, I think I'm a little bit ahead of schedule, but hopefully I've convinced you that these non-canonical amino acids can be accessed by biocatalysis in order to expand to new areas of chemical space, and that biocatalysis enables the efficient, sustainable manufacturing of these non-canonical amino acids. And of course that these really are an important aspect when we're looking into the PMI and sustainability of these peptides. So with that, happy to answer any questions.
Thank you. Thank you, Tina. I don't know if you have any questions, but in the meantime, I will ask you, mostly you have some aromatic amino acids. How is to have some aliphatic amino acid or cycloaliphatic amino acids?
You're asking, do we, or?
Yeah.
Was that your question? So, yeah, we started with the aromatic amino acids since we started with tryptophan synthase, and we can make pretty much any of the aromatic ones at this point, because that's the platform we started with. That's pretty straightforward. We recently added some new flavors of enzymes that do functionally the same carbon-carbon bond-forming reaction, but they just do it in a different sort of catalytic mechanism. And so that's how we've been able to access some of these, alkyl, bioalkyl products as well. But it is the same fundamental idea. We haven't gotten yet to cyclic amino acids. There's no reason why biocatalysis won't be able to do those. Just we started at one end and we haven't quite made it all the way to the other end yet of chemical space.
In other words, you don't see any limitation about your techniques?
So far, obviously every method has pros and cons, but if we're talking about access to chemical diversity, so far we have not encountered one that you just couldn't make. If it sort of meets these underlying principles that would lend itself to a biocatalytic reaction, then it should work. And really it's just a matter of how much work are you willing to put in to do this enzyme engineering, because with just a little bit of enzyme engineering you can access, let's just say, 80% of this chemical space pretty readily, but then there's going to be these little pockets where just the physicochemical properties of those amino acids are a little bit different, and so you may have to rev a little more through this protein engineering. But we've never so far had a product that we really tried to dig in on and just couldn't make at the end of the day.
Okay, we have a question from, let me see, from Raymond. No, I have two questions. Agustin, hi Tina, did you ever run the process in continuous flow?
Good question. So we have done that experiment a little bit, but we've only really dabbled. So the way that we're doing it now is just in batch. We just dump the enzyme into a reactor and call it good, because the efficiency of the reaction and the economics of it are sufficient by doing that. But definitely I am a believer in the enzyme immobilization. That's something that's on our list of process developments in the future. But when we've done it, we're able to immobilize the enzyme pretty easily, but because we're running these at really high concentrations, sometimes the starting materials would just begin to precipitate out. And that's okay in these batch reactions because equilibrium will pull a certain amount into solubility. The enzyme can work on that and then precipitate out the amino acid on the other end. But with the immobilized enzyme, it was starting to get a little bit chunky in a way that we just didn't bother to address yet.
Agustin is also asking about if the enzymes are immobilized.
These ones are not, but they definitely can be. But we do not today.
Okay. Raymond, you say, does side chain precursors have to be water soluble?
No. So, when we do a lot of the tryptophan analogs, for example, indole is extremely insoluble, but then it will just be a giant cake at the bottom of this reactor, and a certain amount of the indole will solubilize into the reaction where the enzyme can see it and work to make the product. And then that equilibrium will just shift. So eventually all of that starting material will solubilize and the product will precipitate out. So that's fine. But when we were just dabbling in immobilization, having that flow proceed with this big insoluble chunks of substrate was just a little bit harder. We didn't try very hard, but that was one of the reasons why we kind of didn't bother at this point so far. But I am very interested in that.
Okay, thank you. And Amika is asking, per kilogram of product, how much enzyme is used and how it is recovered?
Yeah, so the quantities per kg are pretty small. We can do some head math here. Somewhere between, 1 and 50 kgs, I guess. Or no, not per kg, that's per ton. So I guess between like 10 grams, 50, 100 grams to make a kg. We usually talk about it in liters, so sorry, I'm not good at head math, but it's a pretty low catalyst loading, like 0.01%. So we don't recover the enzyme right now. We just let it go because the economics of recovering it just don't make sense for us right now.
Okay, Christian Sorensen from Stanford University. Have you made any non-canonical amino acids with halogens?
All the time, yep. So any of the halogens work at pretty much any position on the amino acid side chain.
And also, do you think that you could engineer an enzyme to make different amines monomers for peptide synthesis?
Yeah, that's not a thing that we've worked much with, but you definitely can use enzyme cascades to make things like, tryptamines, and you can just remove that carboxylic acid as a means to access the amine. We haven't tried to make anything more sophisticated than that.
Okay, Giulio, again from SEM, in amazing, and in the directed evolution, do you have non-natural amino acids in the enzyme itself that can be introduced?
That's a good question. So no, these are just good old-fashioned natural enzymes, and the mutations that we make are all just to natural amino acid substitutions. I am very interested in the incorporation of non-canonical amino acids into active sites, and there have been quite a few good papers on that, where you can change the enzyme reactivity by modifying the active site with these new functional groups. But for us, we've just been doing normal amino acid substitution so far.
Noah Duffy, what are the impurities in this reaction, if you know?
Yeah, so typically the impurities are just residual starting material, or if you're doing a protection, right, some unreacted protecting group. It doesn't generally form things like, beta-alanine. It doesn't form the wrong stereocenter, so we can eliminate those as possible impurities. So yeah, it's usually just unreacted material.
Again, Raymond Yu, how long does it take to direct evolution towards new substrate scope?
Good question. So my answer is it depends. So a lot of times if, let's say, Raymond, you come and say, oh, can you make amino acid A, and it's not one we've made before, we can just run a test on the existing enzymes, because since they have been engineered to be very promiscuous, a lot of times if we just run that test, it turns out one of the current enzymes we know about can make it, and we just didn't know that it could. But if we are starting like truly from scratch, then one cycle here takes 2 to 4 weeks, and you can have to do anywhere between, let's say, 1 and 5 rounds of evolution. So it really does just depend on, when you get a hit that's sufficient for whatever your application is. Do you need to make a tiny bit of amino acid? It's probably a couple weeks. Do you need to make a ton of perfect, high purity product? It's probably a couple months.
Oh, thank you. And also the basis, if you can elaborate a little bit more about the PMI.
So with this figure, what we're doing is, this is the reported value for the peptides. So that's just published in this JOC paper. For us, what we did is we looked here just at the amino acid synthesis. So this doesn't include the FMOC protection or Bach protection, because we were assuming that would be similar across all processes. And so all of these are for 100 kilogram scale. For ours, this is an actual experimental result that we have. So this is using the real yield and everything like that. then these ones we're assuming 100% yield, because while we know what mechanisms are used to make these at scale, I don't have access necessarily to primary batch data from a method that we're not using. So this would be the best case for these methods. Does that answer your question?
Yes, thank you very much. And I move to Hanson for the next presentation. And thank you, Tina. Wonderful work.
Thanks, Tina. So our final talk is from Pascal. He is a team leader in the R&D department at Bachem, where he leads process development projects for both GMP and non-GMP manufacturing. He's going to present a comparison between linear SPPS with SPPS-based fragment conjugation, highlighting the impact on cost and scalability in large-scale peptide production. Pascal?
Thanks for the introduction, and thanks to everyone for hanging out till the bitter end. So I'd like to show you mostly from a cost perspective what we've thought about in terms of linear versus fragment approach in SPPS. And I'll show you a case study too, where we can look at some more evidence-based considerations. Won't bore you for too long with our corporate slide, but I do need to show you really quickly. So Bachem is one of the leading manufacturers of oligonucleotides and peptides. We started off with peptides, got into the oligonucleotide business a few years ago. We're a CDMO, so we do development as well as manufacturing. Ever growing. I heard today that we're aiming to go towards 3,000 colleagues worldwide this year. And that mostly has to do with the ever-increasing demand in peptides at larger volumes. So, as we've heard a couple times in the previous talks, the volumes are increasing. There are more and more, blockbusters on the horizon that are peptidic, and the weight management drugs being the biggest ones on the horizon. And we often get asked by our customers if a fragment-based approach would make more sense than a linear approach. And this certainly, has to do, among other things, with Eli Lilly publishing their approach for tirzepatide, which is a 4-fragment approach, which definitely made this topic a little bit more sexy in recent years. And there are different things to consider. I'm going to talk mostly about cost, but there are many things that could push you in one or the other direction. So, supply chain considerations, capacity, sustainability, PMI. That's not what I want to focus on for this talk here. I really want to look into the cost. So let's look at some calculations. Before I get into that, just want to show you what we mean by a fragment approach versus a linear approach. So in a fragment approach shown on top, you would generate 2 fragments, SPPS. For these calculations, we only looked at SPPS-based approaches. You do a soft cleavage from the resin to have your fully protected fragments. Then you do a conjugation of these 2 fragments in solution, followed by a global TFA deprotection, where you would then end up with your crude peptide, and that would then be purified and isolated in your final steps. In the linear approach, you go directly to the full-length, fully protected peptide, do your global TFA cleavage from the resin while removing your side chain groups, and then you have your crude peptide, and the rest of the downstream is the same for both variants. So, as most of you will know, it really depends on your overall yield per coupling cycle. So that can decrease exponentially, or it does decrease exponentially based on your average yield per cycle. So even at very high yields per cycle, so the top graph here in orange, that's based on a 99% yield for each cycle. You see for a long peptide, you'll have a lower average yield or overall yield in the end, even with a very high yield per cycle. Then if your yield goes down per cycle, obviously this drops off much faster. And the longer your peptide is, the worse it gets. So it can make sense to go for fragmented approach from this perspective. And if you look at just the upstream costs, or for the SPPS, you'll see that while just from a cost perspective, increasing the amount of fragments will be beneficial for your cost calculations the longer your peptide gets. So here we just see for going from 1 to 2 fragments, so that's the dark blue to the light blue graph, and then this kind of goes on. There'll be some inflection point where your process gets cheaper if you go for an increased number of fragments. And if we look at this in a little bit more detail, let's look at 2 metrics. So one is your average yield per cycle, per SPPS cycle, and then your conjugation yield of your 2 fragments. This is just for a 2 fragment, or no, sorry, this is for 1 to 4 fragments. So from a fully linear SPPS to 4 individual fragments based on a 36 mer peptide. And all your other assumptions stay the same for all, for all calculations here. So if both are low, then we don't see a big benefit of going to multiple fragments. You have a low average yield per cycle during your SPPS and the low conjugation yield. The costs pretty much even out in the end. Now, if your conjugation yield is good, but you have a low average yield per cycle, then you start to see a benefit. It's not huge, but you see a benefit going to multiple fragments. If you switch that around, if you have a high average yield per cycle, but a low conjugation yield, so your conjugation gets a little bit less less perfect, then you actually increase your costs by going to multiple fragments. And if both are high, if you have a high average yield per cycle and a high conjugation yield, then again it evens out, but at a lower level, obviously. So this is for a 36 mer. If we look at longer peptides, in the next case we will look at a 48 mer. Then you see a substantial increase in or decrease in costs when you go to multiple fragments. So here in the case that your average SPPS yield per cycle is low, but your conjugation yield is high, then you see a strong benefit in going into multiple fragments. And of course, this gets even more pronounced if your conjugation yield is higher. Then if we flip that around, we see that your linear approach, so just going for one single fragment or not multiple fragments, will start to be more competitive. And if both are high, then yeah, you don't see a huge difference to that. So here the driver is the yield, the average yield per SPPS cycle. Just to summarize this first part, so we see a small effect for short or not super long peptides, 36 mer. You saw there was a little bit of a benefit going to multiple fragments. It becomes a bigger benefit for a longer peptide, especially if your average yield per SPPS cycle is low. So if we kind of tweak those and assume that we've done some process development, but we didn't quite get there on the conjugation, that became less favorable. The SPPS becomes more favorable. Then we'll see that even for a 48 mer peptide, we don't see a huge benefit going to multiple fragments if both yields are low. So the conjugation yield per cycle, the conjugation yield as well as the SPPS yield per cycle. If we increase the conjugation yield, we see a bit of a better effect for multiple fragments. But if we flip that around, of course, that changes. There, quite clearly, the linear synthesis will win. And if both are high, then we don't see a huge difference between all of these scenarios. This is all highly theoretical, based on just calculations. If we sum this all up, the cost advantages can change depending on what your other metrics are. And we really need to, you can't just do this as a paper-based exercise. You need to go into the lab, you need to do some actual tests and, see what your concrete example will yield in the end. And downstream efficiency is something that I didn't talk about as much. Here, the assumption was that the downstream was the same for all, since the process should be the same for all. That can change depending on what impurity profiles you get in your different approaches. So in our case, we went for a feasibility study and looked at a peptide that you'll recognize by its structure as a GLP-1 agonist. This was a 40-mer backbone, 40-mer backbone with a side chain, fatty acid on the side chain. Overall, we had 42 coupling cycles. For this feasibility study, we really only looked at 2 fragment approaches, so not going for more than 2 fragments for 3 or 4. We had to exclude certain conjugation sites, unfortunately, due to IP restrictions. Since it's a GLP-1 agonist, there are already a lot of IP restrictions on it, so some of the best available sites for conjugation weren't doable anymore due to patents. So, for example, we didn't have any glycine coupling sites where we could have done the conjugation, which would have been optimal. Those are already basically taken. So since we had a lot of data on GLP-1 agonist synthesis, just on linear SPPS, we looked at our coupling kinetics and selected 3 sites based on what we knew should, at least on SPPS level, be good coupling sites. This is an assumption that we kind of had to make based on the data we had. And of course, if you wanted to do it completely comprehensively, you would have to actually link the 2 in solution and not just look at the SPPS. So this is, for example, what one of these fragment approaches would look like. So we selected, position 1 here and modeled these 2 fragments, fragment A and fragment B. Then for the linear approach, we did a standard SPPS setup, we used ramage-amide AM linker resin, did our SPPS on resin with a few optimization rounds to get the optimal coupling conditions in terms of time and so forth, and then did a cleavage, standard TFA cleavage, global deprotection, followed by isolation. Then we went for 2-dimensional prep HPLC purification to get to a high purity level, as would be required by kind of our industry standards, and really did a purification then on multi-gram scale to get reliable yields here. And then the final stage was isolation by lyophilization, just under standard conditions, so nothing fancy here. And when we did the fragmented approach, of course, we had to do 2 SPPS on different resins to get the appropriate building blocks. This wasn't the main bulk of the work. Really, the most work had to be put in for this part. So the soft cleavage from the resin, then isolating the fragments was tricky. In some cases, impossible. We had to telescope the solution directly into the next step, so we couldn't do an intermittent isolation. And then the actual conjugation required a lot of fine tuning and screening. Then the cleavage itself was more or less straightforward. Here we just had to remove the side chain protecting groups and then precipitate to isolate the peptide. And the downstream was the same. We used the exact same conditions that we had developed for the linear approach, and made no adaptations to that. That'll be important on one of the next slides that I talk about. And the final isolation was the same, just by lyophilization using the same standard conditions we had used for the linear approach. So, as I mentioned before, and it's really important to highlight, the highest effort by far was this additional step that we had to introduce to get these peptides off the resin, quantitatively, isolate them if possible. And as I said, some cases it wasn't possible to isolate the individual fragments, and then the conjugation itself. And if you ever want to do this at home, I just want to mention it's very important to synthesize the epimer references, because you are going to get a certain amount of epimerization by doing this in solution, unless you have, the absolute perfect conditions, but you need to assume that it's going to happen, unless you can get a glycine, coupling site. And so having these references on hand is really important, because that's one of the key drivers then for the downstream. And I'll show you that on the next slide. So here are the results summed up. So for the fragment approach 1 that we did, we got relatively good, crude purity of the 2 fragments. But when we did the conjugation, we saw 13% epimerization, which is way too much. And we didn't even really bother to purify that, because that especially compared to the other 2 fragment approaches, it just wasn't worth it. Fragment approach 2, we again saw good crude purities, and the conjugation worked well. We got only 2% epimerization, but then for the downstream, when we were purifying it, we actually saw that this epimer increased in the purified material. So it was enriched in the purified material. So it would have required additional development on the downstream process to get rid of that. And again, we weren't interested in performing more optimization in this respect. Then fragment approach 3 that we did, there we got really the kind of the best average crude purities, got very low epimerization, so below 1%, which we could then even separate by prep HPLC. And the overall yield was good. The purity especially was really, really high. So we got over 98% purity in this case. However, when you compare that to the linear SPPS that we did kind of as a benchmark, we saw that this still won basically on every metric. So we got similar purity. The net yield was higher for the upstream as well as the downstream part, and the overall yield was also significantly higher, around 5%-6% than fragment approach 3. And if we calculate back, and this goes back to the previous slides I showed you, so we had an average yield of per SPPS cycle for the linear of above 98%. So we really, with our prior knowledge at Bachem, I think we've gotten really good at SPPS, and we can really, with a bit of optimization, get very high average SPPS yields per cycle, which is then reflected in the overall yield here. So we kind of left it at that, but we did want to think of a couple steps ahead. And so we had some colleagues in-house do some modeling, some cost modeling on this, just using some assumptions that if we were to optimize the 2 processes, so the best fragment approach and the linear approach, with kind of what we would expect to be able to improve. So, increasing your overall yield, being able to reduce your amino acid equivalents, things like that, improve your resin substitution, all these different, numbers and metrics that you can see here. Even after all those potential optimizations, people who did this, financial cost modeling said the linear SPPS would always win, basically. And it wasn't really worth pursuing this fragment approach in this case. So in summary, I mentioned this a couple of times during the talk, you really need a high upstream efficiency, and then your linear SPPS will mostly perform better than a fragment approach, but there's many other things to consider. And this is just from a cost perspective, just to have that disclaimer again. And we really need to do a feasibility study for this type of work, because you're never going to get the actual numbers without testing it in your lab. And it provides a good model for your cost if you wanted to go for large scale manufacturing. And just to say it again, there are many reasons why you wouldn't go for a linear approach. There can be IP reasons. supply chain, it could result in a lower PMI. As Don had mentioned, you know, fragment approach in their case looked to be better. So it's really a case by case basis. But we at Bachem think that, you know, a really well developed linear SPPS, we're going to be competitive if you have a high SPPS yield per cycle. Yeah, and again, the things you can consider are LPPS, where you would need to really have larger volumes. Here, fragment approaches could make more sense, but I just want to warn you, LPPS is something where you need to put a lot more initial development work in, because it's not as standard as SPPS. SPPS, if you want to get to your goal quickly, is really, in our opinion, the way to go. And yeah, I think we've shown that it performs really well. So just as a final acknowledgement, I just wanted to say thanks to the people who helped out here. So my boss, Markus Wiesner. We have Stefan, who did you know, a lot of the cost modeling that I showed in the first part. Matthias, who did the cost projections for larger scale, and then the two teams who worked on the SPPS part and the conjugation part. And thanks to all of you again for hanging in. Happy to take any questions.
Thank you very much, Pascal, for the great. And we start with the first question. Is Zain Tariq, and he says linear SPPS and fragment conjugation will produce structurally different impurities populations, even at the same purity spec. Does the choice of the synthesis route change the clinical risk profile of those impurities?
No, it doesn't change. I mean, you will have different impurities in the different, but I wouldn't say that you have a different clinical risk in the two cases. There's, you know, peptides, I'd say if you know, if you have an epimer of a peptide, I wouldn't expect it to be any more or less toxic than the parent peptide in most cases. That's just me as a chemist speculating. In the end, it really depends how well you can purify, and you will have, you know, potentially a harder time removing certain impurities, especially epimers, that you would see more during the conjugation approach.
Thank you. Waiting for more questions. I thank you for this honest presentation and also highlighting the problem of epimerization. There is a lot of papers right now, lastly, about fragment condensation, patents, and it's very difficult to find comments about epimerization. How serious you think is the epimerization? You showed 3 cases, one with 13%, the second was 2%, and the third one was less than 1%. But how serious you think is epimerization?
I can't give you a general answer to that. I think, you know, it can be very serious. It depends on what kind of coupling conditions you can use for your fragments. That was one of the things that the team that worked on that part of the project was struggling with, was to find the optimal conjugation chemistry for the two fragments that would yield, you know, generate high yield, but then also it'll always be a trade-off with epimerization. So I think you need to design it well. If you have a peptide where you can, you know, select less epimer or non-epimerization prone sites, then a fragment approach would be more attractive. In case you can't, then you're always going to have a certain amount of epimerization, in my opinion.
Okay, thank you. The basis is a great question about aggregation, a peptide aggregation. And the question is, would a fragment approach work better in terms of yield and purity if you have a a prone aggregation peptide?
Yeah, that's definitely a good argument for testing a fragment approach. If you do have a sequence where you've observed or you expect to have, a lot of aggregation during the SPPS stage, if you see that you have a significant yield drop there, that's something that's one of these cases where, maybe you need to redo your cost calculations. And I think a fragment approach could work better, but maybe certain parts of the peptide are going to make it more aggregation prone, and that could be even worse on on the single fragments. It's hard to predict, but I think that's one argument for testing a fragment approach in any case. Yeah.
Okay, John Nestor is asking about costs. From the different equipments, you need multi-synthesizers, personnel, multiple team for fragments, time to completion.
Yeah.
Can you elaborate on the cost of
That's
both
an excellent
strategies?
point. Yeah.
Yes.
So a linear synthesis is always going to be more straightforward, and a fragment-based approach, you're adding extra steps, you're adding extra complexity. You could, envision certain cases where, maybe you can avoid critical impurities, ones that are difficult to separate, and they're, in the end, in the grand scheme of things, you might end up, with a more cost beneficial fragment approach. But in general, I think you need to kind of assume that it's always going to be a little bit more strain on your manufacturing plan if you go for a fragment approach. But if I think Eli Lilly, for example, they do it to kind of guarantee their supply chain, and that's that's also a valid argument.
Okay. I have another question from Elizabeth Denton. She says, at what scale would you start to consider the efforts required for LPPS or TAC-assisted strategies are more cost-effective than solid-phase peptide synthesis?
Yeah, so I think there you really need to go into the direction of tons. And so there are two things to consider. The manufacturing costs there, I find it hard to make an exact prediction, but definitely you're going to have to put a lot more work in to develop a process that'll work with LPPS or tag assisted strategies. Just because it's not as it's not it's not a cookie cutter process like SPPS, where you can use kind of your basic conditions and tweak those a little bit. So I think the main thing is that you need a little bit more patience for the development work and a little bit more of an initial investment. So what could make sense is to go for an initial kind of a first generation approach with SPPS, and then if you see benefits to a fragment based approach, then you could still go for a second generation approach there. But I think I can't put an exact number on it, but I would say you need to be moving in the direction of tons, not just kilos or hundreds of kilos.
And this from myself.
Sure.
What is the advantage? Do you think to use a fragment condensation, but with a tag, will be helpful?
It could be. I especially think that there's a lot of benefits to isolating your fragments, just to have them as clearly defined intermediates. And if you have trouble isolating your peptides or your fragments, or solubilizing them, then a tag could really help. Yeah.
Yeah, because solubility, how you find some real difficult solubility issues of one of these fragments?
Yeah, I mean, some of them we couldn't precipitate at all. They were in this case, too soluble. which was a problem. And it's for commercial manufacturing, you want to be able to characterize your intermediates, and that just works a lot better if you can actually isolate them.
And you say too soluble, but non issues of non-soluble at all?
So they were
DMSO, no DMSO, no DMF, no of this kind of solvents? No, you didn't find any?
so no, they in these cases they were too soluble to precipitate. We couldn't precipitate
Okay.
them well as with our usual anti-solvents. That was the problem. Sorry if that wasn't clear.
Okay.
Yeah.
We have one more question from Charlie. Charlie Johans say you were talking about this long peptide GLP-1, but about cyclic peptide, for instance, Johnson & Johnson peptide was approved last month. This coming from Peptide Dream, from bicyclic therapeutics. What do you think is the strategy to go to a kind of solid phase or fragment condensation?
It's I don't think the structure of the peptide is what drives the decision so much as its size and the exact nature. So there was one where we saw a very nice presentation at TIDES this year, where a smaller cyclic peptide was done in an LPPS approach, using kind of smaller fragments, and that it was just overall what worked best for that case. I can't give a generic answer to that. It doesn't. I don't think it so much depends on whether it's cyclic or linear, but rather kind of on the overall length and what other considerations go into it. Yeah.
Okay, my last question, at least from myself. I
Sure.
was surprised when you mentioned that you are taking advantage to decide the cutting point. You are taking advantage about the solid phase synthesis. This means in the amino acid 13 is a difficult coupling. This also will be a difficult coupling from the fragment condensation in solution. I was surprised, but I want to know your of course, your
That's
opinion.
in the end. That's probably not the perfect approach, but it was the data that we had, right? We have a lot of data on these sequences in SPPS. We know which couplings tend to be more difficult or easier, where we have shorter or longer coupling times. And that was just the best available data that we had, and we had to kind of make an informed guess based on that. It's not ideal. Obviously, I think if you generated all the possible fragments and tried all the possible conjugations, you get different results. In this case, we kind of had to work with what we had on hand.
Thank you very much, Pascal. I move to Hanson for the last 2 minutes. We're on time. Thank you very much.
Thanks.
Yeah, thanks for the presentation, Pascal. Very interesting. Appreciate everyone joining the webinar. We're going to try to host maybe 2 or 3 of these covering peptide synthesis innovations. So maybe 2 or 3 of these per year. So definitely look out for future invites. And Danielle, do you have any other closing comments?
Yeah, so I want to just first thank everyone for attending. If you do not know, we are the Peptide Drug Hunting Consortium. We're a 501c3 tax-exempt nonprofit organization. I've been trying to share my screen, but I don't think it's I cannot share it for some reason. I will be posting this on our website, and I will send a link to everyone once this recording is posted. We also have other webinars that we've done in the past, which are all up there for you to view. And the way we are able to run these webinars is through our sponsors. Without our sponsors, we would not be able to run as an organization. And we appreciate anyone willing to donate and companies that want to help sponsor the Peptide Consortium. And we'll hope to see you at a future webinar.
Okay. Thanks, everyone.
Thank you. Bye-bye.
Bye.
Bye.